slides. So whenever you're ready.
Okay. Thank you. I'm a postdoc in Middle East Technical University, Institute of Marine Sciences. I'm working on a different topic as a postdoc, but what I'm going to present now is recent work that we published in June, and it's a continuation of my master thesis that I did in the same institute. It's about the direct and indirect impacts of harvesting mesopelagic fish on carbon export and some certain food web dynamics in marine ecosystems. I'm guessing that this community is quite familiar with the mesopelagic layer and the mesopelagic fish, but I still want to make a brief introduction just to set the scene for the rest of the presentation. The mesopelagic fish live roughly in between 200 and 1,000 meter depths of the oceans. And now I would like to highlight a few features of them, which are important for this study. The first one is their position in the food web. They have a critical position in the food web because they are feeding on plankton, and they are the prey of top predators such as tunas and sharks. This means that they directly transfer energy produced by the primary producers to the top of the food chain. Another feature of mesopelagic fish is that they do their vertical migration. At nighttime, they go to the surface waters to feed on plankton. They take plankton, or in the context of this study, carbon into bodies in the surface waters. And at daytime, they go down to the mesopelagic layer. They hide from their predators here, and they release carbon back to the seawater here in the ocean interior. So this means that going up and down every day, taking carbon up into their bodies in the surface, but releasing it back to the seawater in mesopelagic layer, they actively transport carbon and contribute to biological pump. Another feature of mesopelagic fish is that they have very high biomass. The estimates vary from 1 to 20 billion tons for global biomass of mesopelagic fish. The variation is quite high, but the consensus is that they are dominating the fish biomass in the ocean. So this high biomass makes mesopelagic fish an attractive target of fisheries. So they are not perfect for direct human consumption because they are tiny. They are tiny and their fat content is quite high, but they can be good sources for aquacultures as fish meal to produce the fish that we like to eat as humans. But the problem is that we don't know the potential consequences of harvesting mesopelagic fish on marine ecosystems because we poorly understand the mesopelagic layer, we poorly understand the mesopelagic fish. We started to understand the importance of mesopelagic fish or their role in marine ecosystems only in the last decade. This is especially because it's very hard to study this region. And there are some studies that estimate metabolic releases of individual mesopelagic fishes. And then this information can be scaled up to the whole community or the biomass that is being harvested to get an understanding of what might be the impact of taking this biomass out of the system. But the thing is that, as I try to explain, they have a very critical position in the food web. They're more or less in the middle of the food web. This means that taking this biomass out of the system is expected to lead more sophisticated changes in the biomass of all groups in the food web. So in this study, we wanted to have a holistic approach to quantify the potential impacts of harvesting mesopelagic fish on carbon export and some other ecosystem dynamics. And for this, we coupled a biogeochemical model with a food web and fisheries model, and ran this model with a mesopelagic fish harvesting scenarios. So the study area is the Sargasso Sea in North Atlantic. There is Bermuda Atlantic Time Series Station in Sargasso Sea, which measures the region for several decades, and this makes Sargasso Sea a favorable place for modeling studies because then we can compare the model results with the data and validate the model. But the other reason why we studied Sargasso Sea was that we had a one-dimensional biogeochemical model which was developed for the region and which was quite good at capturing the carbon export dynamics throughout the water column in the region. So here we have the conceptual diagram of the coupled model, and on the left we have the conceptual diagram of the biogeochemical model that I mentioned that was developed for the North Atlantic. It's called North Atlantic Generic Ecosystem Model. It was already validated and published and used to simulate the carbon export dynamics. So this model is a one-dimensional model. It can represent the 3,000 meter depth of the water column with 3,000 vertical layers. It has detailed structure of algal groups. There are 5 phytoplankton, 2 zooplankton groups, 2 detritus groups with different sinking rates, and dissolved organic matter pools and remineralization of these pools to nutrients and nutrient cycles. What we did in the study was that we added a whole higher trophic level model on top of this lower trophic level model or biogeochemical model. So this higher trophic level model, food web model, is called EcoPath with EcoSeam. It's a very well-known, widely used food web model. We used the foreground version of EcoPath with EcoSeam in order to couple these two models online and in a two-way mode. So we used the North Atlantic setup, EcoPath setup from Vasconcelos 2004. And this EcoPath with EcoSeam works in 2 steps. The first step is EcoPath. So for each group in the system, so in this work we have 31 higher trophic level groups. EcoPath ensures that the mass is conserved. This means that for the given rates, for each group, production minus different mortality terms minus immigration equals to biomass accumulation. So first EcoPath comes, it provides this mass balance for each group, and then EcoSeam takes this output of EcoPath as the initial condition, and it starts to tie dynamic simulations. So in the coupled model, NAGM and EcoSeam are running together simultaneously and providing information to each other at every time step, which was 1 hour. In the end, in the coupled model, we had 42 groups and fisheries. 31 of these groups were high trophic level groups and simulated by EcoSeam, and 11 of them were lower trophic level groups and shown in green here and simulated by NAGM. In the original EcoPath model from Vasconcelos 2004, these groups, which I highlighted in red, were being harvested. So the main target of the fisheries in North Atlantic were tunas, but some other fish groups were being harvested as well. So we use this setup as the reference scenario, base scenario, and ran the model with this scenario without any harvesting of the mesopelagic fish, and then compared the coupled model results with the BATS data. So one of the most critical variables was zooplankton, because in the standalone biogeochemical model, which was validated before, the zooplankton was the end term. It was the highest level of organism in the system. So the entire higher trophic level was represented by the mortality term of the zooplankton, which was constant. So we opened this term up, added the whole food web, and removed the mortality term of the zooplankton to the natural mortality. And then we compared the zooplankton results of the coupled model to make sure that it agrees well with the BATS measurements. And the other important variable is the carbon export. BATS provides carbon export measurements at 150, 200, and 300 meters, and we compared model simulated carbon export at these levels with the measurements from the BATS. And what we also did was to compare the coupled and uncoupled model results against the data, just to make sure that after adding this whole high traffic level on top of biogeochemical model, we didn't, like, model skills didn't go way off from how it was before. So after being convinced that the model is good enough to continue, we prepared mesopelagic fish harvesting scenario. So this was the reference scenario, the original fishery scenario, and we added harvesting of mesopelagics on top of the reference scenario. So we had to decide some harvesting rates, and we first created this baseline scenario. In the baseline scenario, we introduce a fisheries mortality to the system without breaking the mass balance. We wanted to ensure the mass balance without have to change these rates. So keeping the same system, but harvesting the mesopelagics. So we use a very low fisheries scenario for mesopelagics in order to achieve this. But in the second scenario that we called M2 in commercial fisheries, we increased the fisheries mortality of mesopelagics tenfold. So here we are looking at the total biomasses of phytoplankton, zooplankton, mesopelagic fish, and non-mesopelagic fish, and detritus. So what we say that in the reference scenario, the total biomass of mesopelagic fish is more or less 80% of the total biomass of all other fishes. And in the baseline fishing scenario, we don't see much change in the biomasses of these groups. But in the commercial fishing scenario, as expected, we see a significant decrease in the mesopelagic fish biomass, but we also see increase in the total non-mesopelagic fish biomass, total biomass of all other fishes, and we see decrease in the total biomass of phytoplankton. So what happens is that, so here we are looking at the change in biomass in M2 scenario, commercial fishing scenario, with respect to the reference scenario. But we see is that when mesopelagics are being harvested in addition to the usual fisheries, we see strong decrease in the small mesopelagic fish biomass. But when the biomass decreases, the epipelagic fish, which are feeding on plankton and competing with mesopelagic fish for the same resources, are taking the advantage of their competitors not being in the system, and their biomass increases. And we see the highest biomass increase in small epipelagic fish. So then how does this reflect to the carbon export? So in the solid line shows the carbon export profile of the reference scenario. Mesopelagics are not being harvested. And when they're being harvested at a low rate, we see a small shift to the left in the carbon export. So one thing to mention, like in the model, we model the mesopelagic fish in a way that they go to the surface waters at the upper 200 meters at nighttime. They feed on plankton, and their vertical distribution in the water column is distributed in a way that it follows the vertical distribution of the plankton. But at daytime, when they go down to the mesopelagic layer, they're equally distributed to the depths in between 400 and 600 meters. And this metabolic releases of mesopelagic fish here at daytime leads this little bump in the carbon export profile. But in M2 scenario, like high mesopelagic fish harvesting scenario, we see this strong decrease in the carbon export rates, not only in the surface waters, but also in mesopelagic layer. We don't see this bump anymore. We looked at the contribution of these groups to the carbon export. So this is phytoplankton driven, non-mesopelagic fish driven, and mesopelagic fish driven carbon exports. We see that in the surface waters, phytoplankton and non-mesopelagic fish are responsible for the majority of the carbon export. Then they are followed by the mesopelagic fish, and mesopelagic fish contributes to carbon export. In the surface waters, more specifically in the upper 80 meters because their biomass is highest here as they follow the phytoplankton distribution, and then they also contribute to carbon export in the ocean interior. And here we have the change in the relative contribution, like percent contribution of these groups to the carbon export under different harvesting scenarios. So what I want to highlight here is that from reference scenario to M2 scenario, we see the decrease in the contribution of mesopelagic fish to the carbon export, but we see that relative contribution of non-mesopelagic fish increases throughout the water column, and the relative contribution of phytoplankton decreases as well. On the right, we have the same results, but in the form of a table. So we have carbon export rates for different scenarios and for different depth levels, and we have the percent contribution of these groups to the total carbon export. So there are a few points that I want to highlight here. So in the surface waters in the reference scenario, the main controller of the carbon export is the phytoplankton, and then comes the non-mesopelagic fish. And mesopelagic fish, like in the mesopelagic layer, like between 400 and 600 meters, are responsible for the 40% of the carbon export by their vertical migration, active carbon transport. They are almost as important as the passive sinking of particles, like phytoplankton-mediated particles from the surface. But in this commercial fishery scenario, their contribution drops down to 2%, and then passive sinking of particles, phytoplankton-mediated particles, takes the lead in terms of the contribution to the total carbon export. And in the surface waters, with the increasing the epipelagic fish biomass, the contribution of non-mesopelagic fish to the total carbon export it becomes the most important one, and then this is followed by the phytoplankton. We also looked at some fisheries-related ecosystem indexes to see how the system responds to harvesting of mesopelagic fish. The first one is the primary production required to sustain the fisheries. I told earlier that mesopelagic fish directly transfer energy from primary producers to the top of the food chain. And then when they are being harvested, when they are not being harvested, only 7% of the primary production is used for to sustain fisheries, to catch, for the biomass of fish which is being taken out of the system. But in the commercial fishing scenario, it immediately increases to 73%. We lose this amount of fisheries. The second one is the mean trophic level of catch. In the reference scenario, it's 4.08, but with harvesting of mesopelagic fishes, it immediately drops down to 3.2, which is much closer to the primary producers. And the third index is probability of fishes being sustainable. In the reference scenario, this index shows that the system is like from more than 95% being fished in a sustainable way, but this drops down to 20% in commercial fishing scenario. To sum up, the model developed in this study simulates the ecosystem with a holistic perspective, taking the changes in the whole food web into account. Mesopelagic fish do not only have a direct control on carbon dynamics by their metabolic releases and dual vertical migration, but they also have strong indirect controls as being a predator of phyto and zooplankton, and being prey of top predators. The impact of mesopelagic fish on the carbon export varies throughout the water column, and the greatest impact occurs at the time residence depth of the mesopelagic fish, which is in this model set as 400 and 600 meter depths. Harvesting mesopelagic fish leads to reductions in mesopelagic fish mediated carbon export as expected, but it also leads to reductions in the phytoplankton-mediated carbon export, but increases in the zooplankton and non-mesopelagic fish-driven carbon export. Yes, this is the end of my presentation. I'd be happy to take your questions if you have any.
Great. Thank you so much, Deniz. That was really interesting. And I certainly love this cartoon at the end. I'll start off. I do have a couple questions. The first, I just had a question about going back to your carbon export profile slide. Just into, yeah, oh, sorry, that one. Cool. Yeah. So just had a question about what you're tracking here. Is this just POC from fecal pellets and carrion?
Yes, exactly.
Okay.
It's derived from the detritus, like the sinking of the detritus, and then I computed the contribution of each groups to the detrital pool.
Yeah. Okay. Great. So did you account for the DIC from respiration at all in a
we don't have DIC in the system. We don't, like, we only have carbon, like organic carbon.
Yeah. Okay. Yeah. Cool. So there's potentially even more of an importance there, which is interesting. Great. And then the second question I had is, did you run a sensitivity analysis at all? I'm wondering what parameters were the most.
Yes. Yeah, because this is directly derived from the detritus and the sinking velocities. The results are very, like, it's sensitive to the sinking rates of the detritus, this carbon export. And then we use, this is one of the most sensitive parameters that we use. And the other one was, so although the unit of the lower traffic level was carbon, the unit of the high traffic level model was phosphorus. So we needed to convert, we used to carbon to phosphorus ratio while exchanging the information between the models, like this body ratios for fish. And the model is also sensitive to this ratio. What else? Yeah, also the migration depth. So this is predefined as an average daytime residence depth for mesopelagic fish. But if we were to define a narrower residence depth, so this would mean that we would see a like a stronger bump in a smaller region, or vice versa, like it could be the other way around. If we had described a broader daytime residence depth, then we could see per layer a smaller increase in the carbon export rates. What else? Yeah, I think these are the ones that I can say right away.
Okay, great. Thank you so much. Does anyone else have any questions? Oh, yeah, Scott has a question. Go ahead, Scott.
Thanks, Denise. That was a fantastic talk. I was just also interested in the carbon export story when the mesopelagic fish are up in the water column. It doesn't seem to be, they don't seem to be making that big a contribution to the carbon export compared to when they're at depth. Is that because they're doing different things at different times of the day or different depths?
I didn't understand what made you think that they don't contribute much in the surface. So this is their contribution in the surface waters. They contribute. So this one, and then this is. They do exactly the same thing. They go up
I guess it's
surface,
just the proportion in the next figure. The proportions change.
Yes. they do the same things, but they feed here and then
Yeah.
release their metabolic rates here, and then when they go down, they don't feed and then release release their metabolic wastes.
Right. So there's no feeding when they're deep in the water column?
No, because
No.
there is no food source for
No.
them.
Okay. So the assumption is that
we
they're
didn't
feeding.
exactly. We didn't
Yeah.
let them feed on the plankton. We limited their feeding to the nighttime in the surface waters when
Okay.
they can find their prey, which are plankton, and they are in the upper waters, like surface waters, all the
Right.
time.
Cool. And I just had another
But
question. Oh, sorry.
I'm sorry, but there also the small mesopelagic fish, they feed on each other as well. So we let them happen, so they can, because they can find each other deep down, but we didn't let them to feed on phytoplankton. But this also means that for the hours they spend at daytime, down at daytime and up at nighttime, they have different diets in the model,
Right.
because they cannot access the same prey.
Okay.
This might lead to the changes in their releases at daytime and nighttime.
Right. Yeah, and also I just had a question around the fishing pressure. Does the assumption, do you assume that you're fishing throughout the water column, or is it they only fish that so it's all the way through the entire water column?
Yes.
So that 10 times is a full water column integrated fishing pressure, if you like. And if that's the case, does it vary if you modify the way that you fish? Or your depths, the depths that you fish over? Do you think that would have an impact?
It would impact there is a term that defines the vulnerability of fish to fisheries. So
Yeah.
it would this would affect the vulnerability of fish. If it is less vulnerable, then it would impact its biomass and then the entire food web.
And particularly depending on what the fish are doing at the different depths as well, I guess,
Yes,
in terms of what they're contributing to the carbon cycle.
But in this model, like in this particular coupled model, there is no depth dimension for the
Of course.
higher trophic level organisms. So they're being harvested, and we distribute their impact to the seawater, to the water column, to compute the changes in the biogeochemistry. But we don't. So this ecopath in ecosim also has another component called ecospace, where we can define the variations in space. But in this version, we don't have this vertical differentiation in terms of fisheries.
Okay. Oh, no, cool. Thank you.
All right, great. Is there any further questions for Deniz? Doesn't look like it. Well, in that case, we might end here. But I'd like to thank both our speakers, Jean and Deniz, for really fascinating talks that I think really build on each other, looking at different carbon export pathways. So yeah, thank you for everyone who tuned in this week or this month, and stay tuned for our next talks next month.